Hello, hello. Welcome to the Simon Center for Geometry and Physics and to Stony Brook University. My name is John Morgan. I'm the director of the Simon Center. It's a great pleasure to see all of you here today. I think some of you come from high schools who've never been represented here before. So, which high schools do you come from? Which high schools do you come from? Durham Wading River, where the nuclear power plant is. Another one. Smithtown West. How about Smithtown East? Anyone else? Calm down, calm down, calm down. I let them loose. I hope you can control them better than I can. Anybody else that we haven't heard from him? Yes, ma'am. Ward Melville, our local. Any other high schools represented? Islip. How many people from Islip? A couple, okay. All right. So welcome to this edition of the Della Pietra Lecture Series. This is a lecture series generously endowed by the Della Pietra families, and its purpose is to bring scientists to Stony Brook and to the Simon Center. Scientists who are doing exciting, important work that's opening up new avenues in science. That's the first criterion. But also scientists who can explain their science in a way that's comprehensible and engaging to general audiences, like this audience of interested high school students. Well, you're in for a great treat today. We have a home run on both fronts. Professor Bonnie Bassler is a professor of microbiology at Princeton. She has many honors. She's won many honors. She has many awards, which are a tribute to her work. I could list them, or list some of them. Most of them probably wouldn't mean much to you, so maybe I won't go through the entire list, but I will mention that she was a MacArthur Fellow in 2002, was it? This is the Genius Award. Now, in this case, it's probably, well, a good name, but I've certainly known some of the MacArthur Fellows that I wouldn't consider geniuses, but nevertheless, I'm sure some of them are, and I'm sure Bonnie is. She received the World Cultural Council Award for Scientific Merit and the L'Oreal UNESCO Women in Science Award. Maybe more to the point for today's lecture, she won the President's Distinguished Teaching Award at Princeton. She has honorary degrees from several colleges and universities. She's a member of several honorific societies. These are societies whose main purpose is to elect people to honor them. This is the U.S. National Academy of Sciences, this is the Royal Society in the United Kingdom, the American Society of Arts and Sciences. But my favorite of all is in 2010, she was one of the Nifty 50 speakers. These were speakers, I guess, who were chosen because, like for the Della Pietra series, they're doing exciting science and engineering, and they had an ability to explain it, and they gave lectures to middle school and high school students about what they were doing, and Professor Bassler was one of the Nifty 50. So please welcome Bonnie Bassler to talk to us today.
So thanks, John. It is an absolute delight to be here. I'm so glad you guys would come to this talk. And I guess, given the introduction, you can decide either during or after whether you think I'm a genius or not. But I'll not ask you. Anyway, but I'm delighted, really delighted to be here. I love talking to people who think they might want to give science a try. So don't be shy of me, right? If you have a question, yell it out. And so I only have one goal for this talk, and that is to try to convince you that bacteria can talk to each other. And if I manage to do that, my second goal is to try to convince you they're multilingual. And then a sort of sub-goal is to try to convince you that spending one's life working on how bacteria talk to each other is actually practical. That by just doing curiosity-driven science about a question that inspired my imagination many years ago, in fact, that's what scientists do, and those kinds of questions lead to practical applications. And so at the end of my talk, I'm going to try to show you that by learning about bacteria communicating, maybe we can interfere with those conversations to make new kinds of medicines. So that's where this is going. And so I'm going to start just by making sure that we all understand the critter that I'm talking about, which is bacteria. So my first slide has just a picture of a generic bacterium, like an E. coli cell that you would find in your gut. And so most bacteria on Earth look pretty much like this guy. And so what I mean by that is that bacteria are single cells, so they're microscopic. You have to look in a microscope to see one. And they're all put together pretty much like this cartoon shows. And what I mean by that is that they have a membrane. You can think of that as their skin. It keeps the outside world out and the inside world in. And then inside this membrane is the cytoplasm that has all kinds of biomolecules roiling around that allow the bacteria to be alive. So all the things that it needs to make life possible. And then what's special about bacteria is that bacteria only have one piece of DNA. So they have one chromosome that encodes all of the bits of information that it takes to make more of these cells and then to give them all their physical features and their social behaviors. And so what you need to know is that bacteria are the oldest organisms on Earth. So we think that they've been on Earth for about 4 billion years. And they're very simple looking, and because they're so old and simple, for the 400 years that scientists have known about bacteria, they've always thought of them as kind of dumb. And so what I mean by that is if you look at bacteria under a microscope, what you'll see them do is they consume nutrients, so they eat from their environment. They double all of these parts. They double their size. Then they cut themselves in half, and one cell becomes two, two becomes four, and so on and so on. And so it's kind of mundane life, right? They just kind of eat and replicate, eat and replicate. And so because when you just look at them, it looks so simple, and because they have so little DNA, so they probably can't have too many tricks, it's always been thought that they lived as these sort of asocial recluses. And what I mean by that is when a cell divides in half, it has no knowledge, if you will, of its sister cells. They just act as individuals. So that's been the idea until about 20 years ago. So for the first 400 years of studying bacteria. I'm going to try to convince you that it's very different from that they actually chit-chat like crazy, they communicate, and they act in groups. And so in order to motivate you to listen to the rest of this talk, I thought what I would do next, besides just showing you a picture of bacteria, is try to tell you a little bit about your interactions with bacteria in your daily lives. So you already heard that I'm a microbiologist, so I have a very bacterial-centric view of the world, and so I'm going to give you the Bonnie Bassler view of what I think life on Earth is like. Okay, so this picture, what I did is I made a picture of a man, a human, and what I did was I used a calculation, and each one of these circles is supposed to represent some of the human cells in your body, right? And so we know about how many cells it takes to make a human being. There's a few trillion human cells that give you your physical form and then let you do all of the things you do. So that's how many cells it takes to make a human being. What we know is that any time in your life, you have 10 times more bacterial cells in you or on you than you have human cells. So you guys are science buffs, so and you're in a math place, if you do the math, you're 10% human. 90% of your cells are bacterial. But of course, it's not just about the cells. This is the 21st century, and so we know about the molecular biology, and specifically we know about DNA. And so what I've done in this picture is I've remade by human being out of the A's, G's, C's, and T's, the 4 nucleotides that encode the famous double helix of the DNA. And what you guys probably know is that the human genome has been sequenced. So we know how many genes there are in human beings, how many bits of information it takes to make a sophisticated organism. You have about 25,000 or 30,000 genes. So that's how many proteins you make that make you alive. What we know is that you have 100 times more bacterial genes in you and on you than you have human genes. Right? So you can pick either of these metrics. At best, you're 10% human. Really, you're only 1% human. 99% of your genes come from these bacteria. And these are genes that you need because your human genome doesn't encode them. So these bacteria that live in and on you in what we call a biofilm, so they're on your body and in your gut, covering all of your surfaces in this invisible biofilm that's like body armor that keeps the outside world from hurting you. And then what they also do while they're in or on you is do all kinds of functions using these genes that your body can't do. And what we get now is that those functions are essential for life. And so what we now understand is that there would be no life on Earth without bacteria, because every other organism, every animal, every human, every plant, every insect lives in a consortium like this with bacteria, and those bacteria are acting as these 24/7 partners that give you things that you need to be alive, because higher organisms don't encode those pieces of information to make these critical functions. So I hope as you think about yourself going forward after this talk, you're going to realize that you are this community, right? And that's part of your humanness. So now if we look a little closer at these bacteria, what I'm trying to convince you is that what we've learned in the past decade or so is that every other organism on Earth is living in consortium with all of these bacteria that I just told you about. And these bacteria perform all kinds of functions. So some of them that you might have heard about is digesting your food. So if you eat salad, if you eat any plant food, you don't have the enzymes to get the nutrients from those food products. So bacteria digest those and they give you those calories, right? They also provide you with all kinds of micronutrients and vitamins that your own body can't make, and those are essential for being alive. We also now know that these beneficial bacteria that live in and on us play a role hand in hand with your immune system to keep harmful microbes out and to keep good microbes in. So they actually educate your immune system, which is important during development. So this part of the world of bacteria is new. This idea that we're learning that they're actually beneficial to us and they're critical for life. What bacteria have been historically known for is for causing disease. And so these are just pictures under a microscope of a few notorious bacteria that cause diseases that you've probably heard of. And so I don't want to be a Pollyanna here. There are plenty of bacteria out in the world that have no business being in or on you or a plant or an animal. And if they do get a toehold, they can either make you sick or they can kill you. And so now we get the bacteria sort of have two kinds of personalities. There's the bad ones that make us sick, and now these good personalities that keep us healthy. And so we get the bacteria can do both of these things, but the question that my lab has always had isn't that they can do these things, but the question we had is how can they do these things? Bacteria are so small. You need a microscope to see them. So how can it be, if these organisms are these tiny individual organisms, how can it be that they can either keep us alive or they can kill us? Right? So my question is, how do bacteria get any bang for their buck? It seems that individually they'd be too small to make an impact on the environment. So what I want to try to show you is the way bacteria do these things, bad or good, is by talking to each other with a chemical language, counting their numbers, recognizing when they have the right number of cells in the vicinity. If they all do something together, they can accomplish feats that they could never accomplish if they acted as individuals. So they act like enormous multicellular groups, and that's why they're powerful. And so to tell you about that, what I thought I would do today is to just give you a little sort of historical tour of how we got to this idea of bacteria acting as groups, because I thought you might be curious about how you get these ideas and then get more evidence for them. And so what's fun about my field is that this idea of bacteria acting in groups actually came from a sort of anomalous organism, which is shown on this slide. This is the Hawaiian bobtail squid. And the first clue to these bacteria acting in groups actually came from people studying this animal. So this squid is about this big, full grown. It lives off the coast of Hawaii, and it's nocturnal. So during the night, it comes out to hunt, and during the day, it goes to sleep. And this squid lives in a one-to-one beautiful symbiosis with a bacterium named Vibrio fischeri that has the special property that it glows in the dark. So Vibrio fischeri makes bioluminescence, so it makes light like fireflies make light, except the light is blue. And so this squid and this bacterium live together in an association. And so I'm going to tell you about that symbiosis, but first I'm going to tell you about it from the squid's perspective. So what we've done is we've put this squid in an aquarium and the light is on. So it thinks it's day, right? And so the squid looks kind of spastic because remember I told you it's nocturnal. It doesn't like to be out in the day because it could get preyed on. Its predators are out in the day. So it looks really nervous because there's no sediments there. So now what we've done, the same squid, same aquarium, and we've given it sediments, like what it would really find in the ocean. So it thinks it's daylight. It thinks it's supposed to go to sleep. It wants to hide from predators. So you only get eaten by a predator once, right? And so this is a fantastic strategy if your lifestyle is about being out to hunt at night and being asleep during the day and trying to stay away from predators. So now this squid, it made itself invisible from predators, right, by burying itself in the sand. So if I showed you this aquarium, you wouldn't know there's a squid in there. There's actually about 10 of them in there. Okay, so this is the solution. This is the evolutionary solution. During the day, it buries itself just a couple millimeters under the sand. You might have noticed it camouflaged itself. It looks like the sand. It buries itself, and this is fantastic. No predators can find it. The problem is you can't stay there indefinitely. The squid has to come out at night to hunt. It also has to eat, right? So it still has a problem, which is how do I stay invisible from predators at night? This is where the bacterium comes in. So now you're looking at the squid and it's been turned on its back. And I hope what you can see are these two glowing lobes. What the squid has developed is a specialized light organ under the body or the mantle of the squid. And in that light organ, the squid houses this special bacterium, Vibrio fischeri, that makes bioluminescence. So Vibrio fischeri is in this light organ at something like 10 to the 12th cells per milliliter. It's like toothpaste of this bacteria. So they're in there and they're glowing. They're making blue light. And so what happens is that the squid comes out at night and it just lives in sort of shallow, knee-deep water. And so at night, if it was going around hunting, since it's in this shallow water, on bright nights, if there was lots of starlight or moonlight, the light of the stars and the moon can penetrate the depth of the water the squid lives in. So if the squid was swimming around, it would make a shadow that would get projected onto the ocean floor, and predators that were under it could see that shadow, calculate the squid's trajectory, and eat it. So what the squid does is it uses these bacteria in this light organ to counter-illuminate itself. It has sensors on its back, so it can sense how much starlight or moonlight is hitting its back, and then it opens and closes a shutter. It's just the ink sac of the squid that modulates how much light comes out of the bottom, which is made by the bacteria. So it matches how much light hits its back with how much light comes out of the bottom so that it doesn't make a shadow. So it sort of counter-illuminates itself. It's like the stealth bomber of the ocean. It cloaks itself in this invisible device by using the light that the bacteria make to match the starlight. So you guys get it? Yeah. Do you guys like biology? Okay, so that's like the coolest thing in the world. Okay, so anyway, right? Okay, so you get it. So the squid has this symbiosis. It gives this bacterium a home. It feeds the bacteria, and in exchange, they provide light, and the squid uses the light to escape from predators. So that's how it works from the squid's perspective. But what's interesting is that the bacteria only make light at night. They don't make light in the day when the squid is in the sand. And the way the bacteria understand whether or not they're alone, or, whether it's night or day, is by talking to each other. Because what happens is every morning when the sun comes up, the squid buries itself in the sand like you saw, and it pumps out 95% of the bacteria. It doesn't need them. It's asleep in the sand. So now there's only a few bacteria there. It turns out that under that condition, the bacteria don't make light. But then as the day goes by, while the squid is sleeping, the bacteria grow and grow and grow until they get to a high number. And then at night, right when the squid comes out, the bacteria turn on light. So how is it that the bacteria know that they're in those two conditions, whether they're dilute or whether they're together? And what I'll show you is the way the bacteria modulate the light is by talking to each other. So the way this works is as follows. So this now is supposed to be my cartoon of a Vibrio fischeri cell. So in the morning, right, the squid has squirted out most of the bacteria. So now the bacteria are dilute. They're at low cell number. So the world is big and the bacteria are small, even if you're in the light organ of a squid. So when they're alone in the morning, they want to have the program of gene expression going that's good for acting as an individual. So they're carrying out some subset of the number of tasks that they can do. But among the things they do is that they make and release small molecules that we call autoinducers, and you can think of them like hormones. And so what happens is when the bacteria are alone, they make these autoinducers, the autoinducers diffuse away, and that tells the bacteria, you're alone, act as an individual. And in the case of Vibrio fischeri, that means don't make light. So under this condition, the bacteria are dark. But then as they grow and divide, what happens is that since everybody is making its share of that hormone, the autoinducer molecule, the concentration of the autoinducer increases outside of the cells in proportion to cell number. And when this molecule hits a particular amount, the bacteria detect it and they infer by detecting that molecule that they must have neighbors around them. So in unison, when they detect the molecule, all the bacteria turn on light together. And so they make perceivable light, and the squid uses that light. And so what happens is that the squid modulates, this is how the bacteria are in the morning, and this is how the bacteria are at night. And so this just goes on and on and on, so that the bacteria only waste the energy, if you will, of making light right when the squid needs it. And so you understand now how it works for the squid. It keeps this culture fresh by squirting them out every morning. But if you think about it from the bacterium's point of view, they're actually talking to each other. They're telling each other they're there. They're voting with these little chemical votes. They're counting that vote. And then the entire population is going along and doing something together. So this was the first clue that bacteria could have social behavior and could do something in unison. And so to understand how that works, we brought the tools of molecular biology to the table to ask what are the components involved in doing this trick. And so what we found was that there was a gene. So this is now supposed to be my Vibrio fischeri cell. There's a gene and an enzyme. So there's a protein that makes that autoinducer, the hormone molecule. And then that molecule freely diffuses in and out of the cells. So the more cells there are, the more of that molecule there is outside of the cells. So that's how the molecule gets made. And then what we found was that there was a receptor, a protein that sits on the bacterial membrane. This looks just like the receptors on the membranes of your cells. And so it spans the membrane. So it sort of has a part of itself outside of the cell and a part of itself inside of the cell. And so what happens is when the autoinducer, the hormone, builds up in concentration at a particular threshold level, it can lock in like a key. It locks into a slot in this receptor. And then when that happens, the receptor sends information into the cells to turn on the genes that make the enzymes that make light. Right? So it's really simple how this works. The more cells there are, the more the molecule there is at a particular number. All of the bacteria send information into the cell, and in unison, the entire population glows in the dark. And so this was interesting. It showed that bacteria have social behavior. And the reason that it was especially interesting is that it was originally discovered in Vibrio fischeri, right? Because we could just see this bioluminescence that the bacteria made. But then we and lots of other scientists started to study this idea. And what we found was that this was not some crazy anomaly of this bacterium that lives in the ocean. At last count, there are hundreds and hundreds and hundreds of examples of these little circuits in all kinds of bacteria. So now what we get is this ability to talk and to act in groups is common in the bacterial world. And the way bacteria do it is that they have enzymes that make hormone autoinducer molecules. They have partner proteins, and then together these complexes turn on all kinds of genes that underpin collective behavior that allow bacteria to do things when they're in a community, but not when they're alone. And now we have a very fancy name. We're a field, right? And this is called quorum sensing. The bacteria sense when they have a quorum, which is the right number to be able to carry out some task, and then they all do it in unison. So now, again, we probably think that all bacteria, that's a big word, all bacteria haven't been studied, but that it seems to be that these kinds of circuits are all over the bacterial world. So that's how they do it. We also do a lot of chemistry, and so we want to know, well, what are these chemical words that these bacteria are talking with? They're not triangles, they're chemicals. And so the first one that got purified was the one from Vibrio fischeri, because that's the one we've known about the longest. And so it's this molecule. And what you're supposed to notice is that there's a ring and then a side chain, and these are just carbons. But then we and other scientists started looking in all kinds of different quorum-sensing bacteria, and this just shows you a little smattering of different quorum-sensing bacteria and the molecule that each one of them uses to communicate. And what I hope you can see right away is that all of the molecules are related. So they all have this ring on the left-hand side, but then these chains, these carbon chains, are a little bit different in every single species of bacteria. And what these tiny differences do is to confer exquisite species specificities to these languages. So these really do work like locks and keys. You know how all your keys look the same, but they only unlock one door? That's how these molecules work with their receptors. So what I mean by that is if I take the Vibrio fischeri molecule and I put it on Vibrio harveyi, nothing happens. And likewise, Vibrio fischeri is impervious to Vibrio harveyi's molecule. So what we understand is that these molecules confer intraspecies communication. This is how bacteria count their clones, their siblings, their brothers and sisters, and they decide when they and their family members are at a high enough number that they should all do something together. And so I've told you about Vibrio fischeri and how this controls bioluminescence, right? Again, there's hundreds of examples of what these bacteria do as communities. I just put a couple of examples here to try to make the point. So there's a bacterium that's named Pseudomonas originosa. This is just a bacterium that lives out in the dirt. You and I encounter it every day. It's completely harmless to us unless you are immune compromised or you have the disease cystic fibrosis. So you might know that people who have the disease cystic fibrosis, they have a genetic mutation in their lungs. And so they can't clear their lungs. You and I breathe in all kinds of stuff, and we have ways to keep our lungs sterilized. People who have CF can't do that. So they always have a microbial infection in their lungs. What happens is when a person with CF is typically in his or her teens, for reasons that we don't understand, they become permanently colonized by Pseudomonas aeruginosa, and that's what kills people who have CF. So people who have CF die of a Pseudomonas aeruginosa infection, and that's because Pseudomonas has quorum sensing. What happens is the bacterium gets into the people's lungs, it sits in a biofilm, so it adheres to the surface of the lung tissue, it covers itself in this goop, this matrix that makes it resistant to antibiotics, and then as a unit, they all start secreting virulence factors, toxins and proteases, things that chew up the person's lung tissue. All of these things, the formation of biofilms and the group-wide expression of virulence factors, are controlled by quorum sensing. And so this is a terrible disease, but if you think about it from the bacterium's perspective, it's a fantastic strategy. Your immune system evolved to see invading bacteria and get rid of them. So the last thing bacteria want to do, if there's a couple of bacteria in you, is to start secreting toxins and making big biofilms, because those are red flags for the immune system. The better idea from the bacterium's point of view is to wait, to count yourselves and to recognize when you have the right number, that if all of you launch these attacks together, you will be able to overcome the human immune system. So Pseudomonas is a very well-studied example, but there are dozens and dozens of clinically, globally relevant bacteria that must have quorum sensing to be virulent. And so what I mean by that is if we make mutants that can't talk or can't hear, they're not virulent. They have to do this as a coordinated group to make pathogenicity work. Okay, so there's tons of examples of that. I put one more guy here. This one is called Erwinia. This one, well, actually, this one you've definitely encountered. You know how when you guys do eat vegetables, right? You know how when you leave your lettuce in the refrigerator for too long and it turns into that brown soup? You do eat vegetables. You will when you're my age. Anyway, you know how your lettuce and your stuff all turns to soup? That's this guy. This guy is Erwinia. So it's a plant pathogen. And so it's like Pseudomonas. What it does is it infects lettuce or a potato. It waits, it counts itself, and it makes this wound, right, as a collective. But then the really cool and insidious thing this guy does, so besides making these virulence factors, is that simultaneous to making the wound in the plant, it secretes all kinds of antibiotics that it has immunity to. So it makes the wound, and then it kills off all the competitors, so its brothers and sisters can actually get the benefit of the wound, right? And so you get this idea, right? It goes on and on and on. Whenever a bacterium is going to give something away to the world, it's never going to get its own product back. These are called public goods. It's only when the community does it together that everybody benefits. That is quorum sensing. It is how small organisms get to have a big impact on the environment. It's by sharing these kinds of traits. So once we got that far in our studies, we started to think, these bacteria can talk to each other, but most bacteria don't have the privilege of living like Vibrio fischeri in this one-to-one pristine symbiosis with this squid. Most bacteria live like this. This is a picture of human skin. This is what the skin on your elbow would look like. This is a bacterial biofilm. And so what you can see is the skin is covered in bacteria. And every one of these bacteria that's a different shape or size is a different species. And so we in my lab started to wonder if bacteria are really using quorum sensing to count and to do these collective activities, how do they do it if they're in this kind of environment and these languages that we had discovered so far are so species specific? And so we thought, well, perhaps quorum sensing is more complicated than what we had found in the symbiont Vibrio fischeri. And so we wanted to try to explore that question. And so what we did was to use a cousin of Vibrio fischeri, is a bacterium called Vibrio harvii. And so this is a bacterium that we knew about. It's a bioluminescent marine bacterium. It's very closely related to Vibrio fischeri, except it lives free living in the ocean, so not as a symbiont. And so we thought, well, maybe this guy, because it kind of lives in the wild west of the ocean, maybe this guy would have a more complicated quorum sensing system. And so we wanted to study it. And so the reason that I'm showing you this slide, besides the obvious cheap theatrics, right, this is to show you what graduate students do in the lab. They paint on Petri plates. Anyway, no, the reason I'm showing this slide is because I want to try and tell you what the experiment that we did. Right, so this picture, to take this picture, all we did was we turned the lights off in the room, and then we took the picture from the light the bacteria are making. We didn't do anything to them. The light exposed the film. Right, so if we turn the lights off in the room, this is what we see for Vibrio harvii. And so we're geneticists in my lab, and so we thought, well, maybe what we could do is to use this luminescence, again, to find the genes for Vibrio harvii quorum sensing. What we thought we could do was we could just make mutants, just randomly mutate the Vibrio harvii chromosome, plate them out on these Petri plates, and just turn the lights off in the room, and look for bacteria that weren't making light when they should be, or were making light when they shouldn't be. So just by doing that simple trick of turning the lights off in the room and looking for dark and bright cells, we thought we could find the components of the quorum sensing system from Vibrio harvii, right? So it was really easy, a really easy trick, because this bioluminescence makes the invisible world of bacteria visible to scientists. So we did that trick, and sure enough, we found the components. And so what we found was that we found the system that we were expecting. We found an enzyme that makes an autoinducer molecule. I actually showed you that molecule on a couple slides ago. And then there was a partner receptor that detected the autoinducer. So that was like Vibrio fischeri. But what we realized is that when we knocked out the genes for this system, this bacterium still had quorum sensing. So we went hunting again, and what we found was that there was a second quorum sensing system. So there was a second enzyme that made another molecule that I've drawn in pink. It also had its partner receptor, and then together these two systems funnel information into the cell to tell the bacteria to turn on light, and hundreds of other genes, but we're only measuring light in our experiment. So what we realized sort of right away is that there seemed to be two, like a double parallel quorum sensing systems. And so what we wondered when we got that far is, well, why would nature have set it up like this? Why would you have two systems? And what we thought is that if these two molecules don't encode different pieces of information, having two quorum sensing systems isn't better than one. And so we wondered, what could it be that this molecule tells the cells? And so what we did was we cloned and sequenced the gene for this enzyme. And we called it LuxS, for Luxor, the god of light. And so you know how we have the human genome sequence? There are thousands of bacterial genome sequences in the database. So what you can do is you can just take your favorite gene, in our case this gene we had named LuxS, you can take the DNA sequence and plug it into the database, and a computer will tell you, does anybody else have that gene? And so we did that experiment, and what we found is that nearly every bacterium that had been sequenced had a highly conserved LuxS gene. Right? And so I don't know who you like on this page. This is staph, strep, cholera, that's bubonic plague, bad breath, food poisoning, gangrene, all of these terrible pathogens. And this is, for me, I'm like, yum, yum. All these terrible pathogens had this highly conserved gene. And so then we thought, well, hold on a second. These guys are obviously not turning on bioluminescence, right? These bacteria don't have bioluminescence. So what could they be doing with this new molecule? So we and other scientists went hunting for what are the traits that are controlled in these bacteria by this LuxS molecule, what I'll call system 2. And so all these different, this again is just a partial list, all these different bacteria got studied, and in every single case, what we found was just like in the first system, was that biofilm formation, which is the first step in making an infection, and then the release of toxins is controlled by this second molecule. So what we realize now is that there were two molecules playing into these traits. And so then we wondered, well, what is this molecule? How does this molecule work to let bacteria talk to each other? So we purified the molecule from a number of different bacteria, and what we found was that in every case, and now we've done this for hundreds and hundreds of cases, in every case, this is what we call autoinducer 2. These are carbons. It's a 5-carbon molecule, and it doesn't matter what bacterium makes it, they all make exactly the same molecule. So this is fundamentally different than the first language I told you about, where each molecule in each species was slightly different. In this case, all the bacteria use the same molecule to talk to each other. And so what we think this second molecule is sort of a universal language, like the trade language of bacteria, that allows bacteria to talk across species. And so now what we think is that bacteria must at least be bilingual. And what I mean by that is they all have to have some molecule that is specific, that says me, that says you are my absolute identical twin. And then they use this molecule that we call autoinducer 2 to say other. And so the computation that we've actually shown, and we did this recently, that we showed bacteria do, is the first thing we think they do is they're just out there, and what they do is they scan the environment and they just ask, are there any molecules there? Right? And so that tells them you're alone or you're in a community. And then they start turning on group behavior genes based on whether or not they detect molecules. And I've alluded to the fact that in every bacterium that has quorum sensing, something like 200 to 600 genes have to turn on. So it's a big deal to go from acting alone to acting as part of a group. So first they just say, am I alone or am I in a group? And then they start to turn on genes. And then the more sophisticated computation they do is they measure the ratio of these two molecules. And that says, are me and my family winning or losing in any given environment? It says, am I in the minority or the majority, or are you? And then what they do is they change what genes turn on and off based on whether they and their kin are winning or the enemy is. Right? Or somebody else is, right? Because they are out there competing for resources. And so just like we do, they want to know, is it me or is it somebody else? And then they behave differently depending on what they find out. So this is how we think quorum sensing really works out in the world. And there are more molecules to find. There are molecules that say who the other guy is, and people are, like me, are hunting for those molecules right now. But we think that this is sort of the basic skeleton architecture, and that there will be more molecules, but it kind of works like this. You measure the ratios, and that what is encoded in these molecules is something about number and something about how close or how far my neighbor is related to me. And so that's quorum sensing. And then what I wanted to do for the last sort of part of this talk is to try to show you that it's useful to study things like this, right? So I've already told you again and again, right, that quorum sensing controls virulence. It controls biofilm formation and toxin production, right? And so once we learned about quorum sensing, we, the scientists, thought, well, if we could manipulate quorum sensing, maybe we could make new medicines. And you guys probably know that antibiotic resistance is a globally important problem, that we have this terrible problem that there have been no fundamentally new antibiotics for the last 30 or 40 years, and so we're sort of running out of our arsenal, right? And these are molecules that kill bacteria or stop their growth. And so what we wondered is, could you think about antibiotics in a completely new way? And what I mean by that is, instead of killing or slowing the growth of bacteria, which selects for resistance, could you think about behavior modification? Could you make bacteria either deaf or mute, and then they couldn't turn on their virulence genes, and that could be a new kind of medicine. And so we want to try to do that. And so there's sort of two strategies. The first one is, let's say you want what's called a narrow specificity or narrow spectrum antibiotic, right? You have a pseudomonas infection, your doctor wants to treat you for pseudomonas. In that case, you would try to target this intraspecies system, the one that is specific. Lots of times when people get sick, they go to the physician, and the physician doesn't know what you have. And so what you get typically is what's called a broad spectrum antibiotic. You just try to get rid of all the bacteria, right? And so maybe what we could do is to make a broad spectrum anti-quorum sensing molecule by targeting this interspecies, the non-specific system, right? So we're trying both of those strategies, and the logic for how you do it is identical, right? So I'm just going to tell you one story, and it has to do with this intraspecies communication system, but you can use your imagination. We're doing exactly the same thing with this second molecule. Okay, so I've been talking about this guy a lot. So we wanted to try this on a pathogen, right? You can't do this with Vibrio harvii and Vibrio fischeri. They're not pathogens, right? And so we wanted to try it on a pathogen. So we picked this notorious pathogen, Pseudomonas aeruginosa, which is the one I told you about that kills people that have cystic fibrosis. It also infects immune-compromised people, people that have burns. So these are people that already have some problem. And the other really sneaky thing that this guy does is it loves to make biofilm. So it's very sticky. And so it's rampant in hospitals now. Like, you go in to get your gallbladder out, and if they give you a breathing tube or a catheter, this guy gets on those submerged medical devices. And it's becoming like the 19th century again, where you go in for one thing, but you get a bacterial infection, and that's what actually kills you when you go to the hospital. And so this is a real problem in hospitals because it loves to stick to these kinds of surfaces. So the biofilms, which lets it stick to the surfaces, and the toxins, which which is required to make the person sick, are both controlled by quorum sensing. So that's why we picked this bacterium. It has a very well-studied quorum sensing system, and it's a really big problem in the United States today. Okay, so I've just told you this. So Pseudomonas controls biofilms and virulence, just like I told you before, with these quorum sensing autoinducer. It counts, and then it launches. So I showed you on a slide earlier, but you might not recall that the molecule, this is the intraspecies communication system that Pseudomonas uses, is shown here. It's one of these rings with the chain on it, and it actually has two of them. It has one that has this 4-carbon chain, and it has one that has a 12-carbon chain, but they both look like that. So what we did was we made synthetic molecules that look kind of like the autoinducers, but they have a bump on the end. So we made a molecule that slots into the receptor. Like, it goes into the hole for the key, but it sticks there. And what happens is, when this molecule gets into the receptor binding site, these molecules can't get in. Right? So it's an antagonist, it's an inhibitor. And you can see exactly why this might work, right? So here's the real molecule. So this part of our molecule looks a lot like this, but then there's this big group on the end. So what happens is it gets in there, and this big group, sort of plugs the hole. so that the real molecule can't engage the receptor. Okay, so we made that molecule synthetically just by knowing what we know about the real molecules. And so now I want to try to prove to you that it works as an inhibitor. So remember, I told you the first step is the bacteria sit on the surface and then they move out over the surface and they make these biofilm communities. So that's shown on this slide. This is just a Petri plate, and what we did was we just put a little drop of Pseudomonas aeruginosa in the middle of the Petri plate, and then we waited. And it swarms out and it makes this beautiful biofilm pattern as it covers the plate over time. If we do that experiment, but we put our inhibitor in the plate, what you can see is this Pseudomonas can't move. Right? So the inhibitor's in the plate, and so Pseudomonas gets stuck or can't move. And so that's the first positive part of this, is that it looks like this molecule controls biofilm formation. Okay, so that's step one. Step two is the launch of all these virulence factors. Okay, so that's shown on this slide. Pseudomonas is green. I showed you that on the Petri plates. This is just a culture, a high cell density culture of Pseudomonas aeruginosa. The reason Pseudomonas is green colored is because it makes a virulence factor, and this is the molecule that kills human lung cells, that's called piocyanin. For cyan, it makes this virulence factor that is green colored. And so what you can see here in this test tube is wild type Pseudomonas, high cell density. The whole culture's turned green because it's launched and released this virulence factor. If we make a mutant in quorum sensing so that it can't talk, what you can see is that the test tube, the cells are colorless. So that shows you need quorum sensing to express that virulence factor. If we take wild type Pseudomonas, but we squirt in our inhibitor, what you can see is that the bacteria are almost colorless. And so we can quantify this. We can shut that virulence factor down 90%. So that looks pretty good. We can stop biofilms and we can stop virulence, at least in a test tube. So then we wanted to up the stakes and put this in an animal and ask, can we use this to save an animal from a quorum sensing controlled infection? So we did that experiment. So what we measure is how many animals are alive over time. And so if we give a mock infection, right, no infection, of course, all the animals are alive. They'd live for a long time. If we give the animal a Pseudomonas infection, all the animals die in a day. That's because Pseudomonas is doing what Pseudomonas does. So if we do this, but then we add our inhibitor, what you can see is it looks as if we haven't given the infection. So this is the first example of an anti-virulence, anti-quorum sensing molecule that cures an animal of an infection. And so we're actually really happy about this result because we think now perhaps there is merit to this idea of interfering with quorum sensing to make new therapeutics. And so I want to be really clear, this is not a medicine. You saw the molecule. It's one we made synthetically. What one has to do, it doesn't look anything like a medicine. It needs to be much more potent, and medicinal chemists have to build in features that make that molecule look like a medicine. It has to go where we want it to go. It has to not go where we don't want it to go, and it has to be really potent. Right now, you'd have to take a pill like this big and it would kill you. And so we're working on making that look more medicine-like from what we call the lead molecule, which is what I showed you. But we're not there yet. So where we are right now is we're going to take the molecule you saw and we're working really hard to change it, right, to make it look medicine-like. And those tricks are known in the pharmaceutical world. The second thing, remember, Pseudomonas makes these biofilms on these surfaces. So beyond making a medicine, we're now working with engineers. And what we would love to do is to make inherently infection-resistant materials, like biologically inspired materials. So like maybe we can take our anti-quorum sensing molecules and attach them to plastics or metals, right? So you could get an intubation tube or a catheter or a stent that has that's coated with an anti-quorum sensing molecule so that you wouldn't get those kinds of infections. And so that we're doing with the engineers. And then the last thing to say on this, right, is remember is that my lab works on lots of different pathogens, and then there's multiple molecules that we're targeting. So we're doing the same kind of strategies for bacteria that cause other diseases, especially diseases of the underdeveloped world, because we care about those very, very much. And so that's where we're going with the practical stuff. And then to finish my talk, I will tell you everything that I told you in 1 minute. What I hope you believe, so here's the take-homes, is that bacteria can talk to each other. They don't have voices, so they use chemicals as their words. But they're absolutely some kind of communication evolved on this earth billions of years ago. It is not just the purview of higher organisms. What we think quorum sensing does is it lets bacteria be multicellular, right? So again, we believe that multicellularity was invented a long time ago on this earth, that these bacteria have figured out how to do collective behaviors, how to make them robust. And so hopefully by studying quorum sensing in these simple organisms, in bacteria, we're going to learn principles about that other scientists can think about for how higher organisms, like how tissues and cells and tissues in your body work together, or how groups of organisms like animals or people work together or don't work together, as the case may be, and that maybe those principles are embedded in bacteria. With the two molecules I showed you, we think bacteria can distinguish self from other. Again, I would argue that that's what happens in your body and in the world, right? It's not like your kidney cells get all mixed up with your heart cells, and that's because different chemicals tell those groups of cells who they are and what they ought to do. Again, we think by studying simpler systems, we'll be able to help our colleagues that study organs by learning from what these bacteria invented. You know, we came from bacteria, and so maybe the skeletons of the systems that work in our body are actually in these sort of ancient treasure troves of organisms, and that we can learn from that. And it transcends just bacterial research. And then, of course, there's a practical part, which is to develop strategies to impede quorum sensing. But then to go back to the very beginning, remember, most bacteria that we encounter are good. They help us. They keep us alive. What is very understudied, but what is starting to be studied, is how does quorum sensing work in all these beneficial bacteria? So maybe the real way forward in medicine is to stop fighting against the harmful bacteria and actually learn about all these good bacteria that work with us and make pro-quorum sensing molecules to make them talk better to keep us healthy. And so lots of people, my group included, are trying to do that as well. And then the final thing to say is to make a confession, which is we didn't think it up. Once we started thinking about, oh, maybe we could manipulate these systems, we thought, you know, these bacteria have a 4 billion year head start. Maybe they already did it. And so we and lots of other people have been going out into nature, and sure enough, if you look at bacteria that live in these groups together, you find guys that eat each other's autoinducers. So one guy's trying to talk and the other guy eats the signal. So the first guy is like made mute, right? They eavesdrop, they cheat, they make antagonists, you know, right? They make enzymes to cut these signals apart, right? So they're doing all these shenanigans out there. And of course, those strategies have won over billions of years of evolution. So the idea that Bonnie Bassler and some 22-year-old graduate student is going to make a better molecule is ridiculous. And so what we really ought to be doing is just going out there and finding what nature did and using those as the building blocks that we bring to the lab and try to enhance. And so lots of people have a pony in that race as well. And then the final thing that I want to do, I swear this is the final thing, is to show you this is my gang, you know, just a few miles down the road at some party at my house a week or so ago. And what I want to try to show you is, except for that person, they all look pretty close to your age, right? And so what I want to try to just finish by saying is, if any of you are interested in science, it's the greatest life in the world. So remember, my job is to make discoveries. Their job is to make discoveries and to think about things that no one on earth has ever thought about before. We are the 21st century adventurers. And if that's interesting to you, you should really give it a try, even though it's challenging, right? It's really fun. You know, we work in these big groups, we talk, we laugh, we help each other, right? And then what I want to just finally say is that when you hear about new science, like if you read in the newspaper, you hear a podcast or you learn something in school, I guarantee you the person who discovered it is between 20 and 35 years old, right? And so doing science is not a bunch of old white men anymore. These are the people, right, that make the, no offense to my, I only get invited places once. Anyway, these are the people that actually make the discoveries that you read about in the newspaper, right? They come to these labs, they're young, they're a lot like you. It's not some crazy world that you can't be a part of. And so I hope if you're interested that you'll really give it a try, right? And just remember that every idea that I told you about today, somebody in that picture actually did it with his or her hands. Okay, thanks. Sorry.
Can I speak for the species that's about to go extinct?
Sorry.
The old white men. We're not dead yet.
There's plenty of room for all of us to do science. There's so much to discover.
All right, we have time for questions. Who has the first question?
Do bacteria have any strategies to counter your inhibitors?
Absolutely, yes. So I'll say it louder. They have strategies to counter our inhibitors. So they will, right, so you're asking will they get resistant? So they shouldn't come with those strategies because our molecules are synthetic. So they ought not to have seen those molecules out in the world yet, but they will get resistant, right? And so I think we've learned that lesson. You know, when antibiotics first came out, this is right after World War II, it was thought the bacterial problem was solved. And there is no question, the greatest public health measures are clean water and antibiotics, right? So they transform life. But the idea, and nobody had the idea they could get resistant back then, you know, but bacteria do evolution on a 20-minute time scale. So they will get resistant to our molecules. But our fantasy, and this has to be shown, right, is because we're not, so the way a normal antibiotic works is it slows the growth or kills bacteria. So if somebody fortuitously gets a mutation that makes it resistant, it grows, but nobody else does. And that's why you get antibiotic resistance. In our strategy, right, somebody will get fortuitously resistant. And so what will happen, right, so everybody else will not be doing quorum sensing because of the medicine. And the resistant guy will be counting, and it'll say, hey, come on, you know, you guys, let's go. And it will launch its quorum sensing cascade, but nobody else participates, right? So it's like, come on, you guys, come on, you guys. And so in our test experiments, it actually has a growth disadvantage, right? So they will get resistant. It will not be like traditional resistant, which is just impervious to the drug. Our only hope is that these kinds of behavior modifications will give drugs a longer shelf life for you to invent the next thing, right? It's just going to buy us more time than a regular antibiotic because it should take longer for them to get around the strategy. That is the only hope. But I would say just people are targeting all kinds of behaviors. You know, like this, like that's an obvious, you know, like maybe motility or stickiness, you know, and so like you can imagine a cocktail of these kinds of behavior modification drugs, right? And so the idea is to buy time to find the next Achilles heel. Yeah. Really? Oh.
Our immune system has evolved to react on toxins which are produced by bacteria. Why it doesn't react to the autoinducing?
Right. So that's a good question. Yeah. So our immune system, right, recognizes toxins, right? So why doesn't it respond to the autoinducers, right? So toxins, proteins are much, much bigger than like hundreds of times bigger. These autoinducers are really small molecules. And so I want to be careful, though. It has not been done well. Like, are they really invisible to the immune system, or are they really just low level and so they don't elicit a big response? But the idea is they're very small, and so they're not as big of a like, hey, I'm here, as like a toxin would be. Mm-hmm.
When pseudomonas infects someone either through, you know, a hospital infection or like somebody who has cystic fibrosis, and it starts to form the biofilm, what do you imagine is the evolutionary benefit to the bacteria of killing their host?
Right. So you guys hear that? So why does the bacterium want to kill the host? So this is my opinion. I think that they never mean to kill the host. Right? You have 10 to the 14th bacteria in your gut that have evolved to make this mutualism. Like, you need them because you need vitamins and to digest all these things, and they need you because you feed them, right? And so I think when you read about a pathogen, like you read about E. coli 0157, or you read about toxic shock syndrome, what's really clear is that when we, meaning human beings, come smack up against some bacterium that's just out minding its own business, right? And we're a virgin population, right? They have these bells and whistles that are for living out in the environment. And when they get launched on us, right, they overwhelm the immune system, and we, you know, can get sick or die. But that is not in the bacterium's self-interest. The better idea is to evolve together until you become like these mutualists. And so I think that is what's happening. So when you hear about a new pathogen, it's just that it's been out there, and technology changed, the environment changed, transportation changed, right? And then you come in contact and you just haven't had time to beef up your immune system and to work toward this mutualism. I mean, the amazing example is when the Europeans came to the Americas, right? The reason most of us are sitting here is because 90 million Native Americans died of diseases that were now childhood diseases, or the Europeans were immune to them, right? Because they had enough time to evolve together, right? So what I think is 100 years from now, if we were alive, we'd be reading about a completely different set of bacteria, right? They're just out there, and they're never meant to be in you, and they have it kind of off scale, and then over time, you tone it down, right? Yeah, it's a great question. They're all good questions. Anybody else? Shouldn't be shy of me. I mean, obviously. Where? Oh, yeah, good, you're not.
If we are able to make this into medical applications, what do you think is the cost?
Okay, so the question is, if we could make this into a medicine, what would the cost be? It would be high, right? Yeah, so I don't know. I don't know the answer to that. What I can tell you is that an academic doesn't make it into a medicine. It will cost close to $1 billion to make this, to find a molecule that's safe, to do the trials, right? To make sure that it works and that it's safe, right? And so that isn't my job to do. My job is to make ideas. And then my fantasy is if we can get these proof of principles, like the one you're seeing, that a company would pick it up and actually make it. And then what is interesting about antibiotics, so it'll cost a lot to make, but there's a huge need, global, for antibiotics. And what's interesting is that we are used to paying pennies for antibiotics. They're like the cheapest, we've had, right? And so the idea that this would be inexpensive is actually probably correct, because people are used to not paying a lot for antibiotics. And so that's actually I think in my camp, right? And that's the hope. I will not be the person setting those prices if we can do it. Good question. Where? Who? Well, yell it.
Do you think it's possibly linked to the two systems and one of those?
Wait, where are you? Oh, there you are. Sorry. Uh-huh.
Two systems and one of those is probably universal. The other one is very specific. Possibilities that there could be more systems, and biome, and as it gets closer and closer to it, it gets closer, more system common.
Yeah, so I'll say that louder, right? So I showed you a system that says self and other. And so your question is, there a system that says, okay, you're not my brother, but you're my cousin. And then there's a system that says, you're my second cousin, right? So the truth, so we know those exist. So we know there's like two is enough, right, to make the point. There's a molecule that says, I'm a Vibrio. Right? And so the idea that this is speciation. So my colleagues in my field, we believe that. We're brainwashed, of course, right? That the idea, and what you can see it in the molecules. They get longer by 2 carbons, by 4, right? And so that tracks with how close or how far apart these bacteria are related. And so maybe whether or not you can understand one another, maybe that is what it means to be a species, at least in the bacterial world. So that's not proven, but we don't have a good, we use now, you say, okay, if their genomes are 95% identical, that's what we use. I mean, we're much closer to monkeys than that, right? And so that's the wrong, number. It's just what we use. And so there is a lot of idea that maybe as these molecules and receptors evolved hand in hand, and get away from each other, that is what determines your relatedness. I would love that to be true. I don't know how you would prove that, but there's some tantalizing hints, and there are definitely more than 2 molecules. I mean, I need a job, right? So.
You could also try to measure by genetic similarity. Is there any correlation between what you were just describing?
Right. Yeah, so you have to be able to purify the molecules to know what they are, but people are absolutely trying to say, is that tracking with how close my DNA is, like my genome is to yours? Are my molecules, only 1 carbon, 2 carbon, and then how sloppy are my receptors, right? And so that's tomorrow's talk. That is what people are trying to do. Yeah. Anybody else? Just.
There's one down here.
Oh, yeah, sorry.
Would you have to have a different inhibitor for every single kind of bacteria, or is it like a one-size-fits-all?
Yeah, so that's a good question. You hear that you need a different inhibitor for every single bacteria. Is it one size fits all? So the fantasy is if you went after that second system, it'd be one size fits all. And if you went after the first system, it would be a different one for every single bacterium. It will not work that well because those molecules, especially the first system, they're so similar, right, that it's hard to think that there's sort of enough handles to have one size fits all, right? But our molecule, the pseudomonas molecule that I showed you, it doesn't inhibit some other species. So there's parts of it that we think we can make pretty species specific, right? It would be great if we could get each of those things, right? But it doesn't usually work out, obviously, like that. Yeah. Mm-hmm.
The interspecies interaction, once they realize that they're losing an environment, what would they do about it?
Yeah. Right, so you hear that? So like if you, right, so they're measuring the ratios of these two molecules. And so you see a lot of what I'll call autoinducer 2, right? And you say, oh my goodness, that's the enemy, right? What they do is they start to turn on genes so they stop making public goods, right? Like they eat a lot of solid foods and they chop it up into smaller bits that are, easily digestible, right? They stop doing that because they don't want those guys to actually exploit them. And they start making things like antibiotics. They start stealing each other's iron, right? So they sort of make like terrible things, right? So they stop doing the things, the sharing. This is general, right? And then they start actually making defensive products, like you try to kill the neighbor, right? It should sound similar to something you read in the newspaper about what human beings do, right? So it's sort of like that, I think. Yeah, right? So they get less generous, for sure. Mm-hmm.
Made an inhibitor that did affect all bacteria. Wouldn't that also affect the good bacteria you need?
Yeah, that's a great question. So you guys hear that? So let's say we could make this anti-autoinducer 2 molecule, and we gave that's part of your question, it's generic. Right, so wouldn't that affect all the good bacteria, right? I've already made this argument that you need all these good bacteria to keep you alive. And so that's a great question. And the answer is absolutely, it would. And so we are banking on the 80 years that we have had antibiotics. So when you take a traditional antibiotic, tetracycline, chloramphenicol, penicillin, it doesn't just get rid of the bad bacteria. It actually decimates the population of bacteria, for example, that you have in your gut. So that's why you'll get a stomach ache. Women get a yeast infection, right? Because these bacteria get out of whack, right? And so what is so amazing about so the real question is, how did real antibiotics ever work, right? They didn't know about all those bacteria, right? They only thought like if you were sick, you had a bad bacterium. They didn't even know the good ones were there. And so what is amazing that we've learned, the field has learned in the last couple decades, is that you have bacteria that are called persister cells. So when you take a traditional antibiotic and you goof up this, what we call the microbiota that lives in you, you have these pockets of cells that are sort of hidden somehow, and they're immune to the antibiotic. And once you stop taking the antibiotic, those persister cells regrow and they give you back your microflora, which you need to be alive. So we know that from what I would call much more severe traditional antibiotic strategy. So then to your question, if we made this anti-autoinducer 2, what would happen is, sure enough, if we gave it, everybody would be like incapable of talking, but at least they would stop doing lots of the things we need, but at least they would still be there. And then when you finish with the anti-quorum sensing molecule, just like a traditional, at least they don't even have to grow back. They would just start chit-chatting again. That has to be proven, but we're pretty confident of that because of this history of antibiotics being absolutely non-specific, right? So that's why we think this could work.
What's the evolutionary explanation for these pockets of...
Yeah, nobody understands, right? So how do you get these persister cells? Like, who are they? Because when you take them out and look at them, this is like a huge mystery in my field. When you take these persister cells out and look at them, they're not different. They're not genetically different than the bulk of the population. And so nobody understands, right? So you can do this in a flask, and like some very low percent of all the cells are persisters. And so if you take them, you kill off all the good ones, you grow them back, their DNA is identical, right? And so nobody understands. It could be like some stochastic thing where they're in one growth mode and the others are in a different growth mode, but nobody understands persisters. They're cool, right? We need them, and it's a really hot topic.
I guess I was trying to ask, why would they ever be created in the
the first place. We didn't have antibiotics until the last 80 years.
Right. I don't know, but it could be that, but so bacteria make antibiotics, fungi make antibiotics, and so maybe persisters evolved a few billion years ago out in the dirt, right, for something that's similar to what we actually do to them in the host. That is just a wild guess. Right? Anybody else want to quiz me? I'm trying to get them right. Yeah.
So if a bacteria gets its receptor muted and it replicates, will the new bacteria also have its receptor muted?
Yes. So if a bacterium gets a mutation, right, that like makes its receptor impervious to the drug or to the molecule, all of its offspring will also have that mutation. Absolutely. So that's in your DNA. So all you and your kin, right, will have that mutation.
More questions? One last question? If not, let's thank our speaker.
You guys were awesome. Thanks for coming.